I got out of the Army in 1949, so toward the end of 48, I had to start thinking what I was going to do when I got out. I thought of the possibility of going on and really getting my neurology or psychiatry training and becoming a truly qualified neuropsychiatrist. But I still had this lingering feeling that I would like to get back into a research environment. Fortunately, around that time when I was beginning to wonder what I was going to do when I got out, the series of papers in one issue of the Journal of Clinical Investigation came out, papers by my old teachers, C. Marchetti and Carl Schmidt, who were at the University of Pennsylvania. They had developed a method that measured the blood flow, oxygen consumption, glucose utilization, carbon dioxide production of the whole brain in man, and it could be used in the conscious state, which was rather important because, you know, if you use anesthesia, you change the way the nervous system works. And they had developed this method, used it in studying diabetes, hypertension, and a number of other conditions. And there was a series of papers all in one issue of the journal, and I read that, and I said, gee, this is a way of combining my interests. I had developed an interest in the nervous system as a result of the Army experience. I did want to do research because I enjoyed the process of research. And I liked basic research because I liked fundamental questions. And so I thought, well, gee, maybe that's what I want to do. Finally, I saw Dr. Ketty, and I explained to him what I had in mind, and he said, and I had no special ideas, but I just wanted to learn the technique and take part in the program and see where it goes from there. And he said, well, I came at a fortunate time. He had just been notified, I think the day before, that he had a grant to the NIH approved and funded, and he had a salaried position in that grant application with an unnamed investigator, because he hadn't picked anybody yet, and he thought that he would be able to get me into under that grant. But he says he doesn't remember this, but I remember it very clearly. He said actually he had in mind a more experienced person, and the salary was commensurate with a more experienced person, but that he remembered me from medical school, and he would take a chance on me, and he said, and besides, for that amount of salary, I might be able to get two like you. I think there is a tendency for nowadays for people to try to become their own investigators too soon before they're adequately trained. And part of it is the system that I think emphasizes independence, as though somebody who working in a program directed by a more senior person doesn't have the opportunity to exercise independent judgment. And I think maybe people are going off too soon and too quickly into independent projects of their own, which I think impinges on the current problems in biomedical research. My interest in localization of functional activity in the nervous system evolved from my earlier experiences working with Dr. Ketty on blood flow and metabolism of the whole brain. When I came to work with Dr. Ketty, although I had no specific research ideas, I had some questions in my mind. I was interested in the question that I'm still interested in, and that was what happens to the brain chemistry in association with the different functional activities going on in the brain. In particular, because of my experience in psychiatry, I was sort of interested in what happens in the brain and anxiety or panic attacks. The brain is a very heterogeneous organ, not only anatomically, but also functionally. Different parts of the brain are reserved to subserve specific different functions. There's a part for audition, a part for motor activities, for touch, pain, so on. And so we thought what we need is a method that would allow us to measure what's going on in the conscious behaving brain locally, individually, in each region. And preferably, since in many conditions we wanted to study, we wouldn't know where to look in the brain. We'd like to have a method that would look at the whole brain all at once. So we wouldn't miss where to look. Now, I came to work with Dr. Keddy in 1949, and the summer of '51, I guess he left to come here to NIH. So when he came here to NIH, he was scientific director, and his main job was setting up the intramural program in the mental health and neurology institutes. But he had a little laboratory, and he didn't have much help. He had two very bright young neurophysiology fellows who worked with Wade Marshall in the laboratory of neurophysiology, who helped him start a development of a method that would measure local blood flow using that equation, an equation that would allow him to measure blood flow locally. And to get the localization and to measure things conveniently, they used a radioactive gas, which a gas because it diffuses across the blood-brain barrier easily, and they used an I-131 labeled gas, trifluoroiodomethane, which so they could measure the concentration in the blood and in the tissue. And they used an autoradiographic technique to measure local tissue concentrations. And they used an autoradiographic technique to measure local tissue concentrations. I came to NIH in December 1953 and joined that group in the development and implication of and applications of this local blood flow method. And once it was worked out, and we had studied normal conscious animals, the cat in particular, had looked at the effects of anesthesia on local blood flow. We were interested in knowing what happens when you stimulate specific regions of the brain functionally. And so we went to the visual system. Bob Livingston, who was a neurophysiologist who had worked with John Fulton at Yale previously, he had come to the NIH as a visiting scientist, and he worked with us on this. And we stimulated cat's eyes and found spots in the visual systems that got activated on these autoradiograms. And that was the illustration that that was first published in an article that Freigang and Bill Landau, Freigang and I had published. Freigang and Landau, Bill Landau, they were the two fellows who worked with Keddy originally when he first came here. I have been criticized, or I don't know, maybe not criticized, maybe that's too strong, but people have raised questions why I have this obsession with quantification. Well, my background is from really basic science. You know, I was physiology and biochemistry. They measure things. They don't just look at pictures. You know, anatomists look at pictures, but physiologists and biochemists are accustomed to measuring things and dealing with quantitative data. So that's, you know, normal for us. If you can't, as a matter of fact, it was Lord Kelvin who said, if you cannot describe it in numbers, your knowledge is of a meager and unsatisfactory kind. You know? So our methods are all quantitative. After the development of the local blood flow method with trifluoroiodomethane and the use of the quantitative autoradiographic technique in that method, I became interested in using the localization with autoradiography to measure local energy metabolism. My reasons, well, there's several reasons why I was interested in going into energy metabolism. First of all, I did have a tendency to be more interested in the metabolic biochemical processes than in just the simple physical processes of which blood flow is. But there was more than that. Blood flow is a function not of individual cells. There is no cell that has its own private blood flow. If one cell became active, it could cause an increase, a compensatory increase in blood flow by mechanisms that are still being sought. But the blood flow wouldn't be to that one cell. It would be to a whole region involving a lot of cells that weren't activated. So metabolism, which we believe is much, and I think we know now, is much more closely related to the whatever the work of the brain is, the functional activity is. First of all, it's more directly related to it, and secondly, it should give you better spatial resolution because it's more limited to cells. Cells have their own individual metabolism. They don't have their individual blood flow. So we were interested in extending the method to measuring metabolism, thinking that would give us better resolution and better sensitivity for picking up changes in functional activity. That was a problem. I tried first to set up a model, a kinetic model from which you can derive equations, to measure this with a labeled glucose. We knew we couldn't do it with oxygen consumption because the isotopes of oxygen are too short-lived, so we wouldn't be able to get an autoradiographic technique with oxygen. With glucose, possibly, but when I went to do the modeling, I realized that the products of glucose metabolism, carbon dioxide, if you use carbon-14, the label would come off as carbon dioxide, or if you use tritiated glucose, it would come out as tritiated water. These products leave the brain very quickly. And when you measure a chemical reaction, which you're doing when you're measuring metabolism, the best way to do it is to measure the accumulation of product in a given interval of time. And from the amount of product made, you can then determine what the rate was. But if your product isn't staying around, it's like trying to hit a moving target. You don't know how much left. You can't get an accurate measure. So I dropped the glucose approach, and that would be around 1955 or so, 56. In 1957, I was writing a chapter for the Handbook of Physiology on metabolism of the nervous system in vivo. And I was talking with Don Tower, who had a laboratory right across the hall from ours, about what evidence there was that the glucose was absolutely essential for the brain's functional activity. It was the essential substrate, and nothing else would substitute. And one argument was that if you do hypoglycemia with insulin, you lose consciousness, your EEG slows, the oxygen consumption goes down, and if you give glucose, you restore it. If you give insulin and glucose together, you don't get the effects of, you don't get hypoglycemia and you don't lose consciousnesses. And he told me about some work going on in the Heart Institute by Bernie Landau and his associates on 2-deoxyglucose, a compound which, when administered in large doses, produced a picture just like hypoglycemia. It produced coma, but in the presence of a high blood glucose. And they believed that it was blocking glucose metabolism in the brain, and that was responsible for it. The mechanism wasn't known. And so I got interested in 2-deoxyglucose completely for another reason. But so I followed the literature, and then there were some papers that came out. There was one by Wicks and et al., Wick et al., and Bachelard, and Tom Tower had done some work, which indicated that the block of glucose metabolism by deoxyglucose was because the deoxyglucose was being converted to deoxyglucose 6-phosphate and accumulating. It couldn't go any further. And it was accumulating to concentrations that were high enough to compete with glucose 6-phosphate. So the glucose 6-phosphate could not be converted to fructose 6-phosphate. It had blocked it blocked the enzyme at that point by filling the site on the enzyme where glucose 6-phosphate would fit and blocking glucose metabolism. And I remember when I saw that, which would be around, I guess, about 1960 or so, I said, there ought to be some way to use deoxyglucose as a way to trace glucose metabolism, which really was a crazy idea, because whoever would have thought of using a competitive inhibitor of a substrate in a metabolic reaction to measure the rate of the metabolism of the natural substrate. In fact, later on in 1974, when we first reported the deoxyglucose method at the American Society for Neurochemistry, Norm Radin, who was a biochemist, neurochemist from the University of Michigan, had to give some kind of a talk at the banquet of the meeting, and he pointed out, what a crazy idea, and who would have thought it would work? Well, at the time I thought of it, I didn't know if it would work or not, but I thought it was an inkling that there might be a way to do this. And I thought, someday I ought to do something about that. That would be around 1960, 61. I used deoxyglucose in my in vitro biochemical studies for a variety of things, and it worked fine. You could make hexokinase, the enzyme that metabolizes glucose, metabolize deoxyglucose, went to the phosphate, and it was stable. And so I kept thinking, you know, someday I ought to get back to this and do this. But I was busy working on thyroid hormones and protein synthesis, and my laboratory was all attuned to do that. And so you don't disrupt an ongoing program and start another one because you have an idea that could have been a little bit fanciful. But I just kept thinking, someday we have to do this. Well, when Marty Rybich was there and had developed the autoradiographic procedure for carbon-14, now we were able to do autoradiography with carbon-14. And so I remember Kenny Rybich and I used to meet just about every Monday and discuss the research. And I remember raising the point about now that we have a method for carbon-14 autoradiography, maybe it's ought to think about developing the carbon-14 2-deoxyglucose technique. And everybody thought, that's a good idea, you know, but someday we ought to work on it. But nothing was done. And Marty went back to the University of Pennsylvania. I don't remember the exact, I think he went and left in '66. And somewhere not long after that, he called me and he said he's applying for a grant to the NIH, and was I interested in the studies to develop the deoxyglucose technique for measuring local glucose utilization. And I said, sure, I'm interested, but you know, I can't do anything here now. My lab is tied up, and I don't have the space or the people to work on this. He said, well, how about in his lab starting it, and we could work collaboratively, and the experimental work would be done in his lab. Would I be interested? And I said, oh, sure. And so we started in Marty's lab working on the development of it, and did some work with brain slices to show that brain slices use deoxyglucose and glucose in proportion from the medium, and developed a model and an equation which was valid, but it involved simultaneous measurement of blood flow at the same time and solubility of deoxyglucose. So it was theoretically valid, but like so many equations derived from models, it wasn't practical. It was presented at a meeting and published in 1970 by Marty in some book on that meeting for I think the Salzburg Conference book. But it wasn't going any. It was sort of a dead end. In 1968, I decided that I would go to take my sabbatical. We sort of had, it wasn't really a sabbatical, it was a work assignment program. We were allowed to apply for going to another place to work for a year to get refreshment, I guess. And so I decided I would go to France and work in a world-renowned laboratory that was more involved in the biosynthesis of thyroid hormones rather than the actions of thyroid hormones. And I worked with Jacques Nunez, who had done a lot of work on the steps in the biosynthesis of thyroglobulin and thyroid hormones in the thyroid gland. And a critical step in that synthesis was a peroxidase catalyzed iodination of tyrosine residues in the thyroglobulin molecule. And they had a model system where they could, using horseradish peroxidase, could catalyze the iodination of serum albumin or lysozyme or other proteins that had tyrosine residues. What was important was that during that year, doing this modeling of kinetic processes, I became quite facile with enzyme kinetics. And began to think, maybe we should have a fresh approach to the deoxyglucose method that was based on enzyme kinetic principles instead of combination of blood flow and blood-brain barrier transport and so on. And that might lead to a way of analyzing the competition between deoxyglucose and glucose in a way that would lead to a method that we could use to measure local glucose utilization. So when I came back from France, I looked at the state of my laboratory, which was supposed to have been working on thyroid hormones and protein synthesis at the time, and the project was in chaos. It would have taken me months to resurrect it and get it going, and I thought, well, maybe this is sort of a message to change. Maybe we ought to not try to resurrect that project and just start a new project with deoxyglucose. So I called Marty Rivich and I said, I have some ideas about a whole new approach using the enzyme kinetic approach. And is it all right if we can do in our lab? Is it okay to start? He said, oh, sure. So we started working on it. And it worked very, very well, up to a point. We set up the model, we set up the equations, but at the time, we had gotten to the equations this is a very interesting story. We had developed the equations to a point where we had the Michaelis-Menten kinetic constants, Vmax and Km. These are Michaelis-Menten kinetic constants that relate to enzymes of hexokinase for deoxyglucose and for glucose, scattered all over the equation in different places. And we were stuck because it would be impossible to determine the Kms and Vmaxes separately, individually, and each individual part of the brain. It would have not have been practical, and we were stuck. And I guess we were really floundering. At the time, I had a postdoctoral fellow in the laboratory who was supported by province of Quebec. He was a French-Canadian, Michel de Rosier, who was one of the co-authors on the deoxyglucose paper. He had had a very good education in the humanities, not too strong in math or physical sciences, but he was very bright and very compulsive about understanding everything. He couldn't do anything unless he could understand it. And it was wonderful to have him because he forced you to understand it yourself so you could explain it. And he wanted everything explained. Well, one Saturday afternoon, I was sitting in my office, and he came into my office and he said, if you're busy, I can come some other time. I said, no, what do you want? And he said, I have a question. I need explained. I said, oh, what's the question? He said, I don't understand what we're doing and why. And I said, that's a good question. I'm not sure I know either, but I'll explain where we are and what the problem is. So I went to the blackboard and I re-derived the equations up to that point. And I said, if this is where we are stuck, we can't go any further. There's no method. He said, so what are we doing experiments for? What do you expect to happen? I said, well, what we're looking for is some information that would allow us to make an assumption that's reasonable, that would simplify the equation. So we could go further. And he said, I don't understand what you mean. So I said, well, let me give you an example. It's not necessarily true, but I'll make up something that might happen. I said, well, let's look at the equation. And I said, well, you know, we're using tracer doses of deoxyglucose. Tracer theory assumes that the molecular concentration of your tracer is 0. So you could then, wherever you see the concentration of, say, radioactive deoxyglucose, there are many places where you could just say, well, this is essentially 0, because it's being subtracted from another term that's so much bigger that even if it weren't exactly 0, it wouldn't make any difference. So, you know, in fact, that's a very reasonable assumption. And I looked at the equation. There were some places where we could have made that assumption. And I said, well, let's, you know, in fact, that's such a reasonable assumption. I don't know why we haven't made it already. I said, let's make the assumption, see what happens. And when we did that, the whole equation collapsed. All of the Km's and Vmax's came together into one factor as a single factor, and we ended up with an equation that was practical. And by the time I'd gone through, my blackboard was all full of stuff. And when you're doing things on and I'm doing things on a blackboard, you know, I make a lot of mistakes, and you know, add a sign or something like that. So I remember saying to de Rosier, I said, see, this is what we're hoping something like this could happen. This would be fine if it were true, but it's probably not true. I probably made a mistake. It's too good to be true. So he said, oh, now I understand what we're looking for. You know, it was a Saturday. That night I went home, and I had dinner. And then after dinner, I picked up a yellow tablet, and I wanted to know, where did I make that mistake? And I went through the whole derivation again, and it came out the same way. And I did it again and again and again, and I kept looking for my mistake. And I couldn't find it. And by this time, it had gotten after midnight. And I was getting a little excited, but then I thought, well, maybe the problem is I'm making a repetitive mistake. I'll set up the model and use entirely different symbols. So it's not likely I'll make the same mistake again, a repetitive mistake again. And it came out right. And then I began to think, well, does it make any sense? Why should this happen? And then I realized that this combination of constants, which we refer to as the lump constant, was nothing more than a constant that is used by people who do radiochemical enzyme studies all the time without being aware that's what they're doing. It's equivalent to an isotope effect. And I realized that although this wasn't an isotope effect in the literal sense, it wasn't due to the isotope, it was due to the fact that deoxyglucose kinetically was recognized a little differently by the enzyme. Kinetically different, qualitatively the same, quantitatively different. It was the same as correcting, the same way as you correct for an isotope. And it made sense. And that was the big breakthrough. And I'm eternally grateful to Michel de Rosier for that question. The method, the deoxyglucose method, once it was developed, attracted a lot of attention, a lot of favorable attention, because of the visualization aspects of it. But there was a lot of also some hostility that also evolved. As a matter of fact, there was one big controversy about glucose 6-phosphatase, that the deoxyglucose 6-phosphate was not trapped in the tissue, but was broken down and labeled lost. Now, we had considered that when we developed the method, and we looked very carefully and found very little deoxyglucose 6-phosphatase, and in fact, so little that we were able to get a period of up to an hour without having any significant effect of any loss. And we examined it very carefully and showed that. There were some who were wanted to push still the use of radioactive glucose rather than deoxyglucose, and they wanted to make the deoxyglucose method have the same limitations that the glucose method had of loss of product. And maybe I should be careful how I word this, but in some publications that were not what I consider to be the highest quality, they presented false evidence that there was, in fact, significant glucose 6-phosphatase activity. And this really just had a big effect on our program because for some reason, those publications attracted almost as much attention as the original method did. It seemed to be, there seemed to be a lot of people who wanted to see the method destroyed for various reasons. And we spent a lot of time, work done by Tom Nelson and Gerald Dienel in my laboratory, who spent years, actually, I think it was about 5 years of work, proved to find what was it the others did wrong that led them to their conclusions. And some of the things they did wrong are astonishing. I mean, unbelievably wrong. You can't imagine how they could have made mistakes like that. I might say during that period of controversy, people sometimes ask me, well, what have you learned with the deoxyglucose method? And I said, I may not have learned much about the nervous system, but I've learned a great deal about human nature. Deoxyglucose, 2-deoxyglucose, is like glucose in every respect in terms of its chemical structure, except it lacks an oxygen on the second carbon position. It is a substrate for phosphorylation by hexokinase. Quantitatively, it's different than glucose, but qualitatively it's the same. It's converted to its 6-phosphate derivative, deoxyglucose 6-phosphate. Now, as you remember, glucose 6-phosphate is converted to fructose 6-phosphate. For that reaction to occur, there must be an oxygen on the second carbon position. Because 2-deoxyglucose 6-phosphate doesn't have that oxygen, it can't be converted to fructose 6-phosphate. So it's made, and it can't go further down the glycolytic pathway. It can't go to carbon dioxide, can't go to lactate, can't go to carbon dioxide, can't go to water. Now, the question of glucose 6-phosphatase activity was important because if it were broken down back again to deoxyglucose, it could come back out as deoxyglucose from the tissue. And so we looked at that very carefully, and we found that you don't see any evidence of glucose 6-phosphatase activity having any effect on the results until you go beyond 45 minutes after giving a pulse. So that was why we established a method that for 45 minutes. In humans, you can go longer. Now, why is it important to be able to measure all of the product? Well, in a chemical reaction, you want to measure the rate of the reaction. A biochemist, when he measures the rate of a enzymatic chemical reaction, what he does is, at 0 time, he starts the reaction by either, he has all the ingredients necessary for the reaction except one critical one, either the substrate or the enzyme. At 0 time, he adds the last ingredient, starts the reaction, times the reaction, and at a given time, he measures how much product was made. And if he knows all the product that was made, none lost, he measures the total amount of product, he knows how much product was made in a given interval of time. That's the rate of the reaction. He divides the total amount of product by the time interval and so many micromoles of product per minute. That's how you do it in a test tube. And in vivo, it's a little more difficult. That's why the equation is so complicated. But the principle is still the same. You have to be able to measure how much deoxyglucose was phosphorylated over that interval of time, Or if you had used glucose, how much glucose had been phosphorylated. But with glucose, you couldn't tell because it didn't stay. But with deoxyglucose, you could measure how much deoxyglucose 6-phosphate was made. However, there's a problem. If you measured just how much deoxyglucose 6-phosphate was made, you would only be measuring the rate of deoxyglucose metabolism. But that's not what we want to know. We want to know glucose metabolism. So the Lum constant came out of the need to convert from the deoxyglucose phosphorylation rate how much glucose must have been phosphorylated at the same time. The method was first reported in 1974. I gave the theory and the method, and Charles Kennedy, who was with me then, gave the initial results in the rat. And people were interested. It was something new and different and unexpected, but it didn't get very popular. In 1977, the main paper was written in the Journal of Neurochemistry, and it made some impact among the neurochemists, but not too much among the, I think, among the neuroscientists, the general ones. In 1978, the Society for Neuroscience met in St. Louis. And by that time, we had developed a computerized color coding system where we could have the computers solve the equation and convert the autoradiograms into color-coded images where the third dimension was the color. So you had a reproduction of the anatomical structure with the rate of glucose utilization color-coded right in each pixel, you know, right where it occurred. And we first applied it to studies that we did in collaboration with Mort Mishkin's Laboratory of Neuropsychology on animals moving a left hand in response to a visual cue. And these were monkeys in which one hemisphere had been blinded. And the paper was presented by Charlene Jarvis, who's now a council member in the District of Columbia. She was a postdoc with Mort Mishkin. And when she showed these color-coded slides, it created quite a sensation. So the color coding, I think, had a lot to do with it. Also, the paper that showed the ocular dominance columns made a big impact, because although the work of Hubel and Wiesel clearly established that there were ocular dominance columns, their exact anatomy and structure were not really known. In fact, in I think the '68 paper, they had a diagram of what they thought the ocular dominance columns were on the basis of their electrophysiological recording techniques, and they were just rectangles. And this was the first demonstration of the ocular dominance columns in their entirety, just where they were, what they looked like. And in fact, it was in 1973, I was at a meeting in Kyoto, and Torsten Wiesel was there at the meeting, and I had an early slide of an autoradiogram with the deoxyglucose autoradiogram, and I had it with me among my other slides. And I don't even remember what the context was, but there was at one point in the meeting where Bernie Agranoff, who was sitting next to me, said, "Why don't you go out and show him that slide? You know, this may be a look at the future." And so I got up and I said, "We have a method that may allow us to visualize functional activity, although it's in its development stage, and I don't know what'll come of it, but we're hopeful." And I showed the slide. Well, when I got the ocular dominance columns pictures, we made prints of it, And I thought that Torsten Wiesel would be interested in it, since I'd met him at the Kyoto meeting. So I sent him a photograph of the ocular dominance column picture with a note saying, you might find this interesting. Well, about a month went by, and I hadn't heard anything. So I figured, well, maybe he wasn't interested. Then I got a call from him, in which he said sorry, but he'd been away in Europe, and he had just come back, and he'd seen my picture, and he was all excited. And could he send someone down to learn the technique? And he sent us Mike Stryker, who's now at the University of California, who spent I don't know, a week or two with us and learned the technique and brought it back to Boston. And he pointed, and Torsten is a very wonderful person and a very generous person. And I had said, I don't know what we learned with it. It's a nice picture, but I don't know what new we learned. And he said, oh no, you learned something new on this particular study. They had the electrophysiological recordings, which clearly demonstrated there must be something like ocular dominance columns, but they never were able to visualize them. But what they had done was use the axonal transport tracing technique, where they injected radioactive amino acids into the eye or into the geniculate nucleus, and then wait until the labeled protein had moved down to the terminal zones. And they were able to visualize ocular dominance columns in the layer 4 level, but not beyond. Their electrophysiological recordings had suggested that the columns go through the whole thickness of the cortex. Their autoradiographic stuff with the axonal transport tracing technique didn't show that. So they weren't sure, do the columns go through the entire depth of the visual cortex or not? And the oxyglucose method showed it. It went through the entire thickness of the cortex. So he said, that was important for them. So I think it was the combination of that and the color pictures that attracted a lot of attention. Now, once we developed the method, our first goal was to see where could it be useful. First of all, to confirm that it was doing something that correct. You have to check to see that it's doing the right things. So we did what a biochemist would call a recovery experiment. When a biochemist develops an assay for a reaction, the first thing he does is he uses known amounts and sees if he recovers the right amount to check the method. So we wanted to know, is the deoxyglucose method giving us the kind of results you would expect if it were giving us working correctly? So we did experiments where we knew what to expect and did it only to see whether the oxyglucose method could pick it up. Actually, we did that even before the ocular dominance column studies. We started with the rat. We first tried to stimulate the visual system of a rat to see if we could get increases in glucose utilization in visual structures. And we didn't. Until I think it was de Rozier again, who said, well, we're using albino rats. They have their visual system isn't the same. So we decided, well, let's take out one eye, because in the rat, the pathways are crossed about 85%. So we would be able to see a side-to-side difference. So we took out one eye, and sure enough, we were able to see the side-to-side difference in the superior colliculus and the visual cortex and the geniculates. Marty Rivage, who was in the one of our key members of our team that developed the deoxyglucose method, when it was at the University of Pennsylvania, had gone back to the University of Pennsylvania, and he was in the Department of Neurology, and he's a clinical neurologist. And he naturally would like to use a technique like this, like the deoxyglucose method, in human subjects. And we were constant communication all the time. I'd go up there, he'd come down here, and one time he came down here. I remember we were sitting around, Charlie Kennedy and Marty and I were sitting around in my office, and he raised a question about, would it be possible, why could we adapt this for use in man? And I quipped. I said, I didn't see how. Maybe in some countries on special occasions where they still use beheading as a, it might be possible, but I didn't know how you could get the brain out of a man. Besides, I didn't think neurologists would want to have a method where the patient didn't live long enough to pay the bill, you know. And he said, well, at Penn he had met David Kuhl, who was in the Department of Radiology. His field was nuclear medicine. And David Kuhl and his associates had developed a section scanner, they called it, and at that time it was up to the Mark IV section scanner, which was an instrument with 4 scintillation counters placed in the corners of a rectangle, which rotated around a plane of the head and picked up radiation from the tissues in that plane, and with computer techniques could reconstruct images of the concentration of the label at different places in that plane, including the brain. And I think they'd used it already for carbon-11 labeled carbon monoxide to look at local blood volume in the brain. And Marty said, you know, that it might be possible to use that. So the next problem was, okay, if we solve that problem, we still have the problem of getting a gamma-emitting tracer, because the detectors outside the head won't pick up beta radiation. It needs gamma emitters. So we have to get somehow label the carbon of the deoxyglucose. Well, that would have been difficult. Kuhl and Marty brought in Al Wolf, who was a radiochemist at Brookhaven National Laboratories and a wizard at cyclotron use and synthesis of organic radiochemicals. And I remember I came up to Philadelphia, we had a meeting, and I don't remember all the people there. It was Marty, David Kuhl, Joanna Fowler, who worked with, and Al Wolf, who were from Brookhaven, and I think Alavi was there, who was a nuclear medicine man at Penn, and there may have been a couple others sitting in. We were around a big table. We were talking about it, and I had a friend here who had been here, had worked with in the Arthritis Institute, Peter Goldman, who since he's up at Harvard now, was interested in pharmacology and pharmacological chemistry, and he had been interested in fluorinated analogs of natural substances and how they acted biochemically. And he had written a review in Science in which he had pointed out that the fluorine atom is so small. Of all the halides, it's the smallest halide, that if it replaces a hydrogen in a position in the molecule that isn't too critical, it won't alter very much the chemical behavior of the molecule. Like I remember he talked about, he'd made fluoroglutamate, or he had used fluoroglutamate and showed that it formed fluorogaba. So Kuhl's group, Wolf's group, could make fluorodeoxyglucose labeled with fluorine 18. And they fortunately could make it in a 2 position. So we did the necessary biochemical studies here where we showed that 2-fluorodeoxyglucose is just as good a substrate for hexokinase as deoxyglucose. And we injected into animals. We got autoradiograms that looked just like the deoxyglucose autoradiograms. And so it was time to go ahead and do the apply it to use in man. And the first studies were done with the Dave Kuhl's Mark IV scanner, which I guess nowadays you'd call it SPECT, single photon emission computerized tomography. Uh, and the images are not as good as what you get with PET, but it was a beginning. When the fluorodeoxyglucose method came out, it gave a tremendous impetus to the use of PET scanning. In fact, I remember what happened here in NIH. Before NIH had any PET scanning. Don Tower was the director of the Neurology Institute, and Tom Chase was the scientific director. And they came back from a meeting of neurosurgeons in, I think it was in Mexico City, all excited that the NIH has to get a PET scanner. Up until that point, nobody had even thought of getting one here. Why? Well, by that time, the UCLA group had their PET scanning systems working and had been using the fluorodeoxyglucose technique. And there was a neurosurgeon there, Crandall, may still be there, who was interested in the surgical treatment of partial complex epilepsy, where they could localize the epileptic focus, they could remove it and treat it. The problem was localizing it. The way to do it usually was with electroencephalography, including even depth electrodes. But there were a number of patients in which, when there was no seizure, there were no abnormalities in the EEG. But when they had the seizure, the abnormalities showed up, but then they had, it was distributed effects. And so they never couldn't tell for sure where the primary lesion was. Couldn't distinguish from the secondary ones. Well, using the fluorodeoxyglucose technique in such patients at UCLA, they had found that in the non-seizure state, there was an area in the temporal lobe that had very low metabolism, lower than normal, and during the seizure would go way up above normal. So by the combination of what it was like in the resting state and in the seizure state, they could localize it, and they were using it. And Crandall presented this at this meeting, and Tower and Chase had seen this, and they came back all excited, saying, this is the future, you know. This is a new way of looking at how the brain works. That would be very useful. And they were the ones who were responsible for bringing the first PET scanner here to NIH. And so I think there was a relationship between the fluorodeoxyglucose method and PET scanning. It was the first method that gave the PET scanning a purpose, a role, a function. Well, NIH was a wonderful place when I first came. Maybe it wasn't so big, but everybody knew everybody, and there was a lot of space, and we had no problems about budget, our positions and hiring and so on. So everybody cooperated. We used to have a biochemical journal club where I presented, shortly after I came here, the work we had done in human hyperthyroidism, and in which we were surprised to find that in spite of the tremendous increase in whole body energy metabolism, the brain energy metabolism wasn't increased. And there's also, in spite of the fact that hyperthyroid patients have anxiety, and it was a surprise. And out of that came a hypothesis that thyroid hormones weren't affecting brain energy metabolism because they acted primarily on protein synthesis and turnover, and that wasn't very fast in brain. And after that, Seymour Kaufman came to me, and he said, you know, I arrived at the same conclusion from an entirely different direction. I'm a pure biochemist, and it would be nice to test it. Would you like to do that? And I said, well, I'm not really a biochemist. I wouldn't know how to do it. He said, I am. So why don't we collaborate? And in fact, it was with him that I learned my biochemistry. We worked in this on a method for measuring protein synthesis in vitro and studied the effects of thyroid hormones for 15 years or so. Learned a lot of biochemistry, which proved to be valuable later for the deoxyglucose method. That was possible in those days. It's much less possible now, and I don't know why. The climate has changed. And I have a suspicion that it's sort of the phenomenon of these crowded rats that John Calhoun, who used to be at the NIMH, used to study out at, I think it was Poolsville. We had a colony of rats which he allowed to grow in population and get crowded. And they turned from nice, gentle little rats into mean ones as the space got less. But there is now this competition for resources which have become limiting. And so people don't talk as much to each other. I think Dr. Varmus is trying to restore some kind of a collegial atmosphere here. I don't think what he's doing, I think what he's doing is fine. You know, he's having special lectures and meeting groups and trying to get groups of common interest together. But that isn't going to solve the fundamental problem. The problem is to get rid of this constant uncertainty and fear of your resources, of what you're going to have. And it's going to affect the research. You see, when I started on the oxyglucose method, I had no idea it was going to work. In fact, I had strong reservations that we would ever finish it. But this was a place where I could do that and take my chances and take risks and not worry if I failed. That's not true anymore.